Hi, this is Grace Wilmuth, and my partners were Sean Wang and Danielle Legrand. Our video discusses the process of oxidative phosphorylation. This is a metabolic pathway in which ATP is created from a transfer of electrons from NADH or FADH2 to O2 by a series of electron carriers. Simply put, it is a process where the mitochondria uses its structure, enzymes, and energy taken through the oxidation of nutrients to form ATP. This process occurs specifically within the inner mitochondrial membrane. The first part of this process takes place in the electron transport chain. This is made up of a series of interconnected proteins and carrier molecules. This process is the last part of aerobic respiration and is the only part that uses atmospheric oxygen. Electron transport is a series of redox reactions that pass rapidly from one component to the next, to the end point where electrons reduce molecular oxygen, producing water. ATP is synthesized by the end of this step. In the electron transport chain, there are four complexes being used that are embedded in the membrane. The first complex consists of riboflavin mononucleotide, also known as FMN, and an iron-sulfur containing protein. Basically, this is a complex made up of a prosthetic group, which is a non-protein molecule required for protein activity in a protein. This prosthetic group is the FMN. 2 electrons are carried to the first complex aboard NADH. Additionally, this complex pumps 4 hydrogens out of the matrix into the intermembrane space to create the hydrogen ion gradient. The second complex directly receives FADH2, which never passes through complex 1. There are electrons derived from FADH2, which are delivered to the third complex along with the electrons from the first complex. Ubiquinone, or Q, is a compound that connects the first and second complexes to the third by delivering electrons. The third complex is called cytochrome oxidoreductase and is made up of cytochrome proteins in a heme prosthetic group. This heme molecule is similar to the heme in hemoglobin, but it carries electrons instead of oxygen. Cytochrome C is the acceptor of electrons from Q, but it only carries 1 electron at a time. Heme molecules can also only carry 1 electron at a time. Complex 3 pumps protons through the membrane and passes electrons to cytochrome C in order to transport to the fourth complex. The fourth complex is composed of cytochrome C proteins C, A, and A3, and 2 heme groups and 3 copper ions. Cytochromes hold oxygen between the iron and copper ions until oxygen is completely reduced. The reduced oxygen then picks up 2 hydrogen ions from its surroundings to make water. Removal of the hydrogen ions from the system contributes to the ion gradient used in the process of chemiosmosis. Chemiosmosis. This is when the free energy from the series of redox reactions is used to pump hydrogen protons across the membrane. The uneven distribution of positively charged hydrogen ions across the membrane establishes an electrochemical gradient. Hydrogen ions in the matrix space can only pass through the inner mitochondrial membrane through ATP synthase. This is an enzyme that acts as a tiny generator turned by the force of hydrogen ions diffusing through it down their electrochemical gradient. By using the potential energy from the hydrogen ion gradient, the turning parts of the machine are easily facilitated for the addition of phosphate to ADP, which forms ATP. Chemiosmosis is used to generate 90% of ATP made during aerobic glucose catabolism. It is also a method used in the light reactions of photosynthesis to harness the energy of sunlight in the process of photophosphorylation. The overall result of these reactions is the production of ATP from the energy of the electrons moved from the hydrogen atoms. Electrons are used to reduce an oxygen molecule to oxygen ions. Extra electrons on the oxygen attract hydrogen ions from the surrounding medium, and then water is formed. ATP yield. The number of ATP molecules generated from the catabolism of glucose varies. Variance occurs between different species. The shuttle of electrons across the membrane of the mitochondria, and these electrons are picked up on the inside of the mitochondria by either NAD or FAD. The ATP molecules generated from glucose may also be used by intermediate compounds. Thank you. Bye.